Hey everyone, welcome to this online lecture on cell communication, part 1. This chapter was highly requested, so here it is. Please enjoy. This is part 1 of a two-part lecture. The lesson agenda for part 1 is numbers 1 through 3, cell signaling, local and long distance, reception. We will dive into 3 types of plasma membrane receptors and touch on intracellular receptors. If you're looking for the topics in numbers 4 through 7, please see part 2, which I will link in the corner and below. Here are the objectives for this lesson. Please take a moment to look them over with some of the key terms and concepts listed below. Imagine for a moment that you were witness to this scene on the Sahara. How would you feel? What sensations could you perceive? Would you be nervous, excited? Would you want to run? Would you be unable to move? Now imagine that cheetah began to move toward you. How would you feel then? Would your breaths increase, heart pound faster? These physiological functions are all part of the fight or flight response, driven by hormones that are released from the adrenal glands during times of stress. And this is just one small peek into cellular messaging and the role it plays in our ability to be and stay alive. Before we get into the nitty gritty, let's investigate the evolution of cell signaling. Of course, this always leads us to microorganisms. Ancestral signaling molecules likely evolved in prokaryotic and single celled eukaryotes. We've studied how prokaryotic bacterial cells can signal to each other. Some coordinate actions to form large complexes called biofilms. Pictured here is a scanning electron micrograph of a mixed culture biofilm. A biofilm is an aggregation of bacterial cells adhered to the surface. Another example is the secretion of toxins by infectious bacteria. They do this to remove competing organisms. Quorum sensing is a special type of cell signaling that allows bacteria to sense local population density. Interfering with the signaling pathways used in quorum sensing may be a promising approach as an alternative to antibiotic treatment for antibiotic resistant bacteria. Yeast cells also signal each other to find other yeast cells for reproduction. Molecular details of signal transduction in yeast and mammals are actually pretty similar. And interesting to note, at best, you are about 10% human, 90% bacterial cells. Some estimates put us closer to 1% human, depending on which metrics you are looking at. When talking about forms of cell communication, we have to make the distinction between intra and intercellular. These root words are derived from Latin. Intra means inside. So, intracellular communication is within the cell. In this picture of an animal cell, you can imagine all the signaling that is required within to keep the cell alive. Inter means between. So, intercellular communication is between two different cells. In this video, the two neurons are sending signaling molecules called neurotransmitters between each other. Now, let's review some signaling basics. As we go through this lesson, you'll see these terms used over and over as we flesh out the details of cell signaling. First, the signal. Chemical signals are released by signaling cells in the form of small, usually volatile or soluble molecules. Another term for a chemical signal is a ligand. Examples of ligands are hormones, ions, simple sugars, and neurotransmitters. In the image, you can see the orange stars and the blue squares as different types of ligands. A ligand is a molecule that binds another specific molecule, often delivering a signal in the process. We are, after all, talking about cell signaling, right? Ligands can thus be thought of as signaling molecules. Ligands interact with proteins in the target cells, which are cells that are affected by the chemical signals. These proteins that ligands interact with are called receptors. There are many types of ligands and receptors, but always keep in mind that specificity is crucial in cellular biology. And so, a specific ligand will have a specific receptor that typically will only bind that ligand. Just think of them like puzzle pieces. There are 4 categories of chemical signaling in multicellular organisms: autocrine, paracrine, junctions, and endocrine. 3 are local. They would be autocrine, paracrine, and junctions, while endocrine is long distance signaling. So let's go over each. Autocrine is when a cell targets itself. The prefix auto meaning self. This is when a signaling cell can bind to the ligand that it released. In this image, you see the cell releasing that red ligand, which then turns around and binds the cell's own surface. There are many instances in the body where autocrine signaling is utilized. Autocrine signaling regulates pain sensation and inflammatory response. If a cell is infected with a virus, the cell can signal itself to undergo programmed cell death called apoptosis or apoptosis, which we will talk about in part 2. It also often occurs during the early development of an organism to ensure that cells develop into the correct tissues and take on proper function. Signals that act locally between different cells that are close together are called paracrine signals. Para means beside. So a signaling cell is targeting a nearby target cell. Paracrine signals move by diffusion through the extracellular matrix. This means that the ligands flow from high concentration to low concentration down its electrochemical gradient. These types of signals are usually quick. They elicit quick responses that last a short period of time. To keep that response localized so it doesn't diffuse too far, paracrine ligand molecules are quickly degraded by enzymes or they're removed by neighboring cells. Removing that signal, that ligand, is going to help reestablish the concentration gradient so that the next time the signal is released, it can quickly diffuse through that intercellular space and find its target. Examples are growth factors, which are usually seen as capital G, capital F. An example is IGF-1, insulin-like growth factor 1, which stimulate nearby target cells to grow and divide. One example of paracrine signaling is synaptic signaling, the transfer of signals across synapses between nerve cells. That junction that's between 2 nerve cells where that signal, the neurotransmitter, is sent is called a synapse, hence the name synaptic signaling. So again, here the nerve cells release chemical ligands called neurotransmitters. The small distance that's between those nerve cells allows the signal to travel quickly, which enables an immediate response, such as if you put your hand on a hot stove and want to move it. And drugs that are used to treat depression and anxiety and PTSD are often affecting these synaptic signaling processes. Junctions are examples of Local direct cell signaling. Here, a signaling cell targets an adjacent target cell connected by cell junctions. One such junction is the gap junction, which is found in animal cells. This is called local direct because those two cells are in direct contact. Both animal and plant cells have cell junctions that directly connect the cytoplasm of the adjacent cells. Gap junctions, again, are in animal cells, and plasmodesmata are in plant cells. You can see a picture here of plant cells. They're kind of rectangular shaped. Remember, they have a more sturdy structure than animal cells, and the plasmodesmata will be the junctions between all of those cells. Gap junctions and plasmodesmata are fluid-filled channels, and they allow small signaling molecules or ions such as calcium to diffuse between the two cells, but large molecules like proteins and DNA cannot fit through these channels. And just remember, they are fluid-filled, which will be mostly water. So anything that's flowing between there also has to be water soluble, like ions. Cell surface molecules are another example of local direct signaling. In the image, you can see the two cells have cell surface molecules attached to them in their plasma membrane, and they're reaching out and contacting each other as a form of cellular communication. These are important in embryonic development, immune response, and maintaining adult stem cell populations. We now move on to long distance signaling. In endocrine signaling, a cell targets a distant target cell through the bloodstream. This comes from Greek endo means within and crine means secrete. Endocrine signals originate from endocrine cells that are usually from endocrine glands. Can you think of an example of some endocrine glands? We have the gonads, we have thyroid gland, and pancreas as a couple of examples. The ligands that are released in endocrine signaling are called hormones. You've all heard of hormones. Hormones travel long distances between endocrine signaling cells and the target cell. And again, the highway that they're able to move through is the bloodstream. The ability of a cell to respond to a hormone depends on whether it has a receptor specific to that hormone. Again, remember, blood reaches nearly all cells in the body via the vasculature. If a signal, aka a hormone, is flowing through the bloodstream, we only want effects on certain cells. Hormones produce a slower response as opposed to some of the other types of chemical signaling that we just saw. They are lower in concentration and are dilute, moving through the bloodstream, and have a longer lasting effect. So that concludes the 4 categories of chemical signaling, and it was a lot of new info. If you've watched my other lectures, you know that I'm a huge proponent of mind maps. Getting all the concepts onto one page where you can see them in relation to one another is a game changer, especially for exam prep. So if you're not already doing this, give it a try. This is what I put together for cell signaling types. On the top left, we have long distance and details about this kind. On the right, you see local signaling and some details there. Under long distance, in the yellow box, there's endocrine and details about it, including the name of the ligand, where it's released, and examples of where endocrine signaling is used. On the right, in the red box, we have local signaling, 2 types of direct and 2 types of non-direct. Stemming from each type are examples. For cell surface molecules, we see they are utilized in stem cell population, immunity, and development. For paracrine, I listed growth factors, that they're often signaled this way, and synaptic as a type of paracrine signal. I honestly think that one of the most important skills you can learn in high school or college is organizing all of this information. For me, this always allowed easier recall and application. So if you're interested in a copy of the schematic, look in the description box below. Also, we're about halfway through this part 1 of this lecture. So if you have a moment, I'd love if you would drop a comment. Let me know what works for you for online studying, what doesn't work for you, and any topics that you have a hard time with that you think other students would also benefit from a video like this. Now we will move on to these stages of cell signaling. This is the mechanism of what is occurring. There are 3 stages: reception, transduction, and response. For the rest of this lecture, for part 1, we will be in the yellow reception box. Here we will discuss intracellular and transmembrane receptors and their mechanisms. If you're looking for details about transduction or response, please see part 2 of this lecture. If you're looking at this and it's confusing, no worries. By the time you're done with my lecture, you will be familiar with everything on this schematic. Again, if you'd like a copy, see the description below. So here we are again, the 3 stages of cell signaling: reception, transduction, response. In reception, a target cell detects a signaling molecule that binds to a receptor protein on the cell surface, or as we'll see, intracellular. In transduction, binding of the signaling molecule alters the receptor and initiates a signal transduction pathway. These pathways have many names: transduction pathway, signaling cascade, secondary messenger cascade. These are all synonymous for the series of steps that leads to a response. I like to think of this part like dominoes. Once the first falls, the rest will follow. Finally, there is the response. Here, the transduced signal triggers specific response in the target cell. This can be gene expression, growth, metabolism, or cell death. For the rest of this lecture, again, we will focus on types of receptors utilized in step 1, reception. Here they are, the 4 types of receptors divided into 2 categories. We have intracellular receptors, just on the left, and then the 3 on the right are cell surface, aka transmembrane receptors. Within the transmembrane category, there are 3 types of receptors. We see ion channel, enzyme RTKs, and GPCR. Let's begin with intracellular receptors. Remember that the prefix intra means within. So these receptors are found inside the cell, notably the cytoplasm and the nucleus. In this image, the receptors are the blue triangles, the ligands are the red circles, and are hydrophobic, meaning water fearing. Now, why do the ligands need to be hydrophobic? Because the plasma membrane and the nuclear envelope are both made of phospholipid bilayer. Phospholipids are hydrophobic in the center and would only allow a small hydrophobic ligand to pass through. Anything that is hydrophilic, water loving, will be blocked from crossing the membrane. So, those blue intracellular receptors in the cytoplasm and the nucleus are binding small hydrophobic ligands, pictured as the red circle. When they cross the nuclear envelope, they find the DNA and they bind to that DNA to activate the transcription of various genes. We can therefore call them transcription factors. And this is how they regulate gene expression. A transcription factor is anything that will aggregate onto the DNA to help kickstart transcription. And just as a reminder, transcription is when we read the DNA to make mRNA. Now we move on to cell surface receptors. Cell surface receptors are also known as transmembrane receptors, are obviously on the cell surface. They are membrane anchored integral proteins, and their job is to bind external ligand molecules. Trans means across, so this type of receptor spans the plasma membrane. There's some in the extracellular area, it goes through the membrane, and there's some in the intracellular area. These receptors perform signal transduction through which an external and extracellular signal is converted into an intracellular signal. Ligands that are interacting with cell surface receptors do not have to enter the cell that they affect. On the right, you'll see each cell surface receptor has three main components. There's an external ligand binding domain called the extracellular domain. There's a hydrophobic membrane spanning region called the transmembrane domain, and an intracellular domain inside the cell. Now, because cell surface receptor proteins are fundamental to normal cell functioning, it should come as no surprise that a malfunction in any one of these proteins would have severe consequences. Errors in protein structures of certain receptor molecules have been shown to play a role in hypertension, which is high blood pressure, asthma, heart disease, and cancer. The first type of cell surface receptor is the ion channel. Ion channels are gated, which means that they need to be activated to open. And we can do this with voltage change, which would be the flow of positively or negatively charged ions to the area, or ligand binding. Once these channels are open, they allow for the controlled flow of ions into and out of the cell. In figure A, the channel proteins remain closed until a signaling molecule shown as the orange teardrop binds to the channel protein. As seen in figure B, the channel protein then undergoes a conformational change, meaning it changes its shape and allows the ions, those yellow circles, to flow into or out of the cell. When the signaling molecule is released, the channel protein will resume its closed conformation, its closed shape, preventing ions from flowing, as seen in figure C. Ion channels allow ions such as sodium, calcium, magnesium, and hydrogen to pass through. Each ion channel is specific for which ion it will allow to pass. These channels are important in action potentials of neurons, muscle contraction, such as cardiac muscle in the heart, and certain toxins, such as bee stings, take advantage of affecting ion channels. The second type of cell surface receptor is a type of enzyme. Let's focus on kinases for just a moment. A kinase is an enzyme that catalyzes the transfer of phosphate groups from ATP, which is adenosine triphosphate. We can see that the two purple spheres with three phosphate groups. So it catalyzes the transfer of phosphate groups from ATP to another protein. So kinases are responsible for phosphorylation. Phosphorylation. Whenever you hear that word, know that there's a kinase involved. In this image, we can see the ATP dephosphorylated to ADP, which is adenosine diphosphate. It now only has two phosphates. And where did that third go? We can see the green protein is phosphorylated with that third phosphate. If the enzyme is a tyrosine kinase, the phosphate group is added onto the amino acid tyrosine. There are also kinases that specifically phosphorylate other amino acids like threonine and serine. So remember, enzymes are always specific for the substrates that they will catalyze reactions for, and they're usually named after them. So again, in this example, we're talking about the receptor tyrosine kinase. So we will see phosphorylation of tyrosines here. So here we have the receptor tyrosine kinases, or RTKs. Receptor tyrosine kinases are membrane receptors that catalyze the transfer of the phosphate groups from ATP to another protein, which again we call phosphorylation. You can think of phosphorylation as an on-off switch because this usually activates or deactivates a protein. A receptor tyrosine kinase can trigger multiple signal transduction pathways at once, and abnormal functioning of RTKs plays a role in oncogenesis. It is associated with many types of cancers. So notice in the picture on the left, the receptor tyrosine kinase, or RTK. Many receptor tyrosine kinases have the structure that's depicted schematically here. Before the signaling molecule binds, the receptors exist as individual units that we refer to as monomers. Notice that each monomer has an extracellular ligand binding site, an alpha helix spanning the membrane, and an intracellular tail containing multiple tyrosine. Right now, these are inactive monomers. The binding of a signaling molecule, like a growth factor, to that extracellular domain causes two of the receptor monomers to associate closely to each other. And this forms a complex known as a dimer, di meaning two. And this process is called dimerization, or we can say that the two receptors have dimerized. Dimerization activates the tyrosine kinase region of each monomer, so the enzyme part of the monomer. Each tyrosine kinase adds a phosphate from an ATP molecule to a tyrosine, which is part of the tail of the opposite monomer. This, of course, requires energy, ATP. We see here that 6 ATP were utilized, and we have 6 ADP as a result. This process is called autophosphorylation or cross-phosphorylation because the left monomer is phosphorylating the right monomer, and the right monomer is phosphorylating the left monomer. Now that the receptor is fully activated, the phosphorylated residues can transmit the signal to the next messenger within the cytoplasm. So here's specific relay proteins, which are shown in purple inside the cell, will bind to a specific phosphorylated tyrosine. The tyrosine in turn phosphorylates that purple relay protein, causing a structural change that activates that bound relay protein. Remember, phosphorylation often activates or deactivates proteins. So that's what's happening here. It's causing a structural change in that purple relay protein, which is activating it. Each activated protein then can trigger a transduction pathway, leading to cellular response. I mentioned earlier that RTKs play a role in oncogenesis. In 30% of human breast cancers, HER2, which is human epidural growth factor receptor 2, is permanently activated, resulting in unregulated cell division. There are certain drugs used to treat breast cancer that inhibit HER2 receptor tyrosine kinase autophosphorylation. The third and final cell surface receptor is the GPCR or G protein coupled receptor. GPCRs are the largest family of cell surface receptors. They work with the help of a G protein, as we will see shortly. Here, water soluble signal molecules, which cannot move through the plasma membrane because they're hydrophilic, bind to specific sites on receptor proteins that transmit information from the extracellular environment to the inside of the cell. GPCR systems are extremely widespread and diverse in their functions. Here's a better look at the receptor. There are 7 transmembrane domains, H1 through H7. This is one continuous protein. Notice the amino terminal in the top left, NH3+, and the carboxyl terminal in the bottom right, the COO-. The transmembrane domains are alpha helices made of hydrophobic amino acids. Here we see the G proteins associated with the receptor. G proteins are all very similar in structure. This is a heterotrimeric G protein, meaning 3 different proteins. Hetero meaning different, tri meaning 3. We have alpha, beta, and gamma subunits. Activating this receptor can be thought, again, as an on-off switch. So this happens when G proteins bind an energy-rich molecule called GTP. Notice that this energy molecule is slightly different from ATP. ATP is adenosine triphosphate, while GTP is guanosine triphosphate. And you can see GTP pictured in the bottom right corner over here. Let's walk through this mechanism. The G protein starts loosely attached but able to move along the cytoplasmic side of the membrane. A G protein functions as a molecular switch, which is either on or off depending on whether GDP or GTP is attached. When GDP is bound to the G protein, which is shown here, the G protein is inactive. Once GTP is bound to the G protein is active. The receptor and G protein work together with another protein, usually an enzyme. Once the appropriate signaling molecules bind to the extracellular side of the receptor, which is shown in green, is activated, and this causes it to change its shape. So there's a conformational shape change. The cytoplasmic side of the receptor then binds an inactive G protein. Again, the inactive G proteins have GDP. But once it binds, this causes the GTP to replace the GDP on that alpha subunit. And this activates the G protein. The activated G protein disassociates from the green receptor, and the beta and gamma subunits also dissociate from the alpha subunit. The alpha subunit diffuses along the membrane and finds the next target in this transduction pathway, which again leads to a cellular response. This is the beginning of signaling cascades, which again we will cover more in part 2. Note that binding of signaling molecules is reversible. Like other ligands, they bind and they dissociate many times. The ligand concentration that's sitting outside the cell near the receptor determines how often a ligand is bound and initiates signaling. Obviously, if there's a higher concentration of ligand, there's going to be more signaling. Hydrolysis of GTP to GDP terminates the signal, and the G protein is once again inactive. The GTPase function of that G protein allows the pathway to shut down rapidly when the signaling molecule is no longer present. GTPase is an enzyme. Anything that ends in ase is an enzyme. GTPase is the enzyme that allows the alpha subunit to break up GTP to GDP and a phosphate. So we've completed the first box, the yellow box, reception. When you look at this schematic, you should now be familiar with the terms and how they relate to one another. And in part 2, we'll move through transduction and response. If these videos help, please let me know in the comments below. I'd love to know what online materials you're looking for, what you can't find, and works best for you. Thank you so much for watching. I hope you're rocking your classes, and I'll see you next lecture.